signals are those which are going to give the information regarding physiological system, human physiological system. The biomedical signals are going to give the information regarding human physiological system. Actually, they are going to give what is the health condition of the system, what is the health condition of biomedical, the human physiological system. So it is very important to extract the biomedical signals. It's a very extraction of the biomedical signal is very important, which is used for the diagnosis of various diseases. Okay, traditionally, actually, the biomedical signals are analyzed in time by the skilled physicians. But now here we can use a number of signal processing techniques can be used to analyze the biomedical signals and to extract the relevant information from the signals and to remove the unwanted information such as artifacts, noise, all those things can be removed by using the various signal processing techniques. Okay, coming to the types of biomedical signals: electroencephalogram, EEG, electrocardiogram, ECG, electromyogram, EMG, electrooculogram, EOG, electroretinogram, ERG, electrogastrogram, that is EGG. Electroencephalogram is going to give the functioning of the brain. Actually, to obtain this electroencephalogram, the electrodes actually are going to measure the potentials from the various parts of the body to obtain the biomedical signals. So here, to obtain the electroencephalogram, we are going to place the electrodes on the scalp, so that we are going to obtain the we are going to measure the potentials from which we can obtain the working of brain, functioning of the brain. Okay, similarly, electrocardiogram. So where the electrodes are placed on the chest, so that we can obtain the potentials from the various locations, so that we can obtain the functioning of the heart. And electromyogram, which is used to obtain the functioning of the muscles, muscle contraction and all those things can be obtained by using electromyogram, where electrodes are connected to the various muscle positions. Okay, next, electrooculogram and electroretinogram, both are corresponding to the retina functions. And electrogastrogram is going to give the functioning of the various nerves within the stomach. Okay, these are the various types of biomedical signals. And I'm going to discuss about ECG and EEG in this session. Okay, so this is the figure that is going to give the cardiac system. This is the cardiac system. Actually, here this is the the heart can be divided into four chambers. That is upper two chambers and lower two chambers. The upper two chambers are called atriums, and lower two chambers are called ventricles. Lower two chambers are called ventricles. Okay, so the upper two chambers, the right, we are going to have right and left. So actually, here the upper, that is right, the right. So the right atrium is going to collect the. It is going to collect the upper two chambers, that is atriums, are going to collect the blood. The right one is going to collect the blood from the veins from the body, that is which is rich in carbon dioxide. That is, it is carbon. It is with the carbon dioxide. And the left atrium, this one, the left atrium is going to collect the blood from the lungs, which is rich in oxygen. Okay, and similarly, the lower two are the ventricles. So the right ventricle is going to, and these two ventricles are going to pump the blood out of this heart. So the right ventricle pumps the blood to the lungs, which is rich in carbon dioxide, and the left the left ventricle is going to pump the blood to the various parts of the body through the arteries, and which is rich in oxygen. Okay, so actually here initially, if we consider this first right atrium, the functioning of the right atrium, so it is going to collect the blood here. It is going to collect the blood from the veins, which is rich in carbon dioxide. Okay, and actually after collecting the blood here, after a while, this portion, the portion of the two atriums will be get compressed. The portion of the two atriums will be get compressed, and then the blood in the atrium will be pumped down. When the atriums are compressed, this blood will be pumped down to the ventricles. Okay, and when the ventricles receive the blood, actually here the flow will be only in one direction. There are some valves which are going to allow the flow only in one direction. That is from the upper chamber to the lower chamber. Okay, when after the pumping the blood into these ventricles, now the atrium becomes relaxed. Atriums becomes relaxed, and then when the ventricles receive the blood, and then after receiving the blood, after that while again these two atriums, these two ventricles get compressed. When these two ventricles compressed, the blood in the right ventricle will be supplied to the lungs, and the blood in the left ventricle is supplied to the body, various parts of the body. So the main thing is, in one time, the upper two atriums will be compressed. First, they are going to collect the blood, and they will become, whenever the two atriums are compressed, the blood will be released to the lower ventricles. Okay, so actually the upper two atriums are going to collect the blood. The right is going to collect the blood from the that is veins from the body, and the left is going to collect the blood from the lungs. The right is rich in carbon dioxide. The left portion is going to collect the blood from the lungs, which is oxygenated. Okay, and when it is compressed, the both will be pumped to the downwards to the right, the right atrium to the right ventricle, and left atrium to the left ventricle. Okay, when this ventricles is compressed, the blood in the right atrium will be supplied to the lungs, which is carbon dioxide, which is rich in carbon dioxide, and the left ventricle is going to supply the blood to the various parts of the body. So actually, all these operations are to be synchronized in proper way. So actually here there will be some nips which are going to produce the potentials. There are some nips which are going to produce the potentials. If you observe this sinus node, here the nips in this sinus node are going to produce the potential. So that potential will be spread to the walls of this atrium. The potential will be spread to the walls of the atrium. Because of that potential only, the compression of this atrium takes place. Because of the generated potential, the near sinus node, which will be spread towards to the entire part through the walls of the atrium. Okay, and this way the potential will be spread to the Beckmann's bundle. Okay, and because of this potential, whenever the potential is released and because of its spread, the compression in this atrium takes place. The compression in the atrium takes place. Okay, and then here, when there is a compression in the atrium, the blood will be flowed downward. The blood in the atrium will be pumped downwards. Okay, so at that time, actually here there is a node called AV node. There is a node called AV node which acts as like a buffer, and it is going to delay that the signal. It is going to delay the signal that is produced in the sinus node. It is going to delay that signal, so that within the delay time, what happens that is the blood will be, blood in the atrium will be pumped to the ventricles. Okay, the blood in the atrium is pumped to the ventricles during the delay time. So after that delay, this AV node is going to release the potential that is generated in the sinus node. It is going to release the potential through this bundle of His. See here, this is a bundle of His. Through this bundle of His, the potential will be released. The AV node is going to release the potential to this bundle of His, and then here it will be divided into two paths. One is left bundle branch, the other is right bundle branch. Here it is divided into two parts. Okay, so that it is a left bundle branch and right bundle branch. And again, if you observe here, for each bundle of branches, there are some fibers which are connected. That is said to be Purkinje fibers. They are called Purkinje fibers, and which are acts as a nips. So that because of this Purkinje fibers which acts as a nips, the signal that is action, the potential that is received here, that will be spread over these complete ventricles at a time. The signal here received by this AV node, that will be spread to the entire ventricles at a time because of this Purkinje fibers which acts as nips. That is unlike to that what is the in the case of atrium. In the case of atrium, the signal generates the sinus node that spreads slowly toward the entire atrium. So the compression of the atrium is not at a time. The compression of atrium is slowly from one portion to other portion, it will be compressed. But here, because of this Purkinje fibers, and that is the potential will be distributed at a time, so that all these ventricles compress at a time. So that because of when this all these ventricles compress at a time, what happens, that is the blood in these ventricles will be pumped out. The blood in the ventricles will be pumped out, so that the right ventricle pumps the blood towards the lungs, okay, which is rich in carbon dioxide, and the left ventricle pumps the blood to the body, which is rich in oxygen. Okay, so these are the potentials. If you observe what is the potential that we can observe, and what is the related to these activities, if you observe this figure, which is in right side, the figure in the right side, see here. So here, whenever the potential is generated at sinus node, we are going to have this shape P wave. This P wave is produced whenever the potential is generated at this sinus node. So that when the P wave is produced, what happens? There is a compression of this atrium. So that the blood will be pumped to the ventricles, that is downwards. And there is some gap between and P and Q. There is in this portion we are going to have straight line. There is no signal in this. There is no wave in this portion. So actually here it is a delay. Actually here this AV node is going to produce some delay, within which time the ventricles are filled with blood. The ventricles are filled with blood. So in this delay time, after that, when the AV node is going to release that blood, we are going to have the propagation of the count. That is the signal will be reached to the entire part. The potential will be reached to the entire part, and then the compression of this ventricles takes place. Okay, and this compression of ventricles takes place at a time here. So we are going to have large peak like this, QRS. So we are going to have large peak R because of compression of entire ventricles at a time. Okay, and this is the large peak that we are going to obtain in this cardiac system, that is in this activity, cardiac activity. And then again we are going to have some straight line portion here. The straight line portion is because of here it will be in that position for some time, and after that you are going to have T. So the T corresponds after polarization. This has to, after compression, again they have to be relaxed. After compression of the ventricles, again they are going to get relaxed. That is said to be depolarization. Because of the depolarization, we are going to have the T wave. So the P wave corresponds to compression of atrium, and the R wave, R corresponds to the peak, highest peak R corresponds to the compression of ventricles, and the T wave corresponds to the depolarization of ventricles. So similar to the depolarization of ventricles, how we are going to get the T here, we are also going to have the wave corresponding to the depolarization of the atrium also. But what is the thing is, here we are going to have the peak R, so that the depolarization wave corresponding to the atrium will be mixed with that one. So as compared with this large peak, we are unable to observe that small peak. Because of large peak at R, we are unable to observe the depolarized wave corresponding to the atrium. So these are the peaks which correspond to the electrocardiogram, P, Q, R, S, T. And there is a lot of importance for this observation of this waveform, depending on what is the peak values and what is the time instant at which we are going to have this one. So here we are going to find the various amplitude values for P, R, and Q, all those things, and we are going to find what is the latency. That is what is the time period. Okay, what is the time period? Actually, we are going to have the PR segment, this period, and we are going to have QT segment. We are going to have various segments. Among that, this QRS is a very small signal. If you observe this QRS segment, that is very short in duration, And if you observe, all these waves are going to have very short duration compared with the one complete cycle. Actually, this is going to give one complete cycle. Again, you are going to have the same cycle will be repeated again. Okay. And here, if you observe, in this way, you are going to have the peaks repeatedly. These cycles will be repeated periodically. So actually, here, what is the rate that is usual rate is, it is in the range of 72 like that. Okay. So actually, we are going to consider for the analysis, we are going to consider what is the gap between R and the peak values. That is, we are going to consider RR interval. RR interval means what is the peak, R peak, that is uptime, and then what is the gap between the two corresponding R peaks. So why are considering the R peaks means why? Because there is a maximum value, that is the large peak. So we are going to consider the duration between the two R values. Actually, if it is what is the abnormalities means, if that is less than 60, if that is the number of the number of peaks, the number of peaks per minute. If you observe the number of peaks per minute, if it is less than 60, then it is said to be bradycardia. So this number of cycles will be less than 60 means what it implies, that is oxygen and supply of oxygen and glucose will be in shortage to the various parts of the body. So if it is less than 60, there is a shortage of oxygen and other glucose to the various parts of the body. And if it is greater than 100, if this there is a rate is greater than 100, that corresponds to the tachycardia. So where it is functioning more, the heart is functioning more. So it is also, it should not be like that. If the heart is functioning more, then sometimes it become tired and then completely it will not work. So that is called failure of heart. The other is the sinus arrhythmia. So actually here, if you observe this signal, it is like a periodic signal, but not exactly periodic signal. This ECG signal is not exactly periodic signal. We are going to have some deviation. If this deviation is greater than 10% from one cycle to the other cycle, from one cycle to the other cycle, if the deviation is greater than 10%, it is said to be sinus arrhythmia. Okay. So actually, this analysis of this PQRS wave is very important regarding the amplitude and time instance. Okay. And this is the picture that is going to give the connection of the electrodes, electrode positions on the body. Okay, next coming to the EEG, so actually it is electroencephalogram. Electroencephalogram. So actually the electrodes are placed on the scalp. Okay, electrodes are placed on the scalp. Actually, this system is said to be 10-20 electrode placement system. 10-20 electrode placement system. Actually, this 10 and 20 is going to give what is the distance that is measured in percentage from nasayan to inion. So that is nasayan means we are going to have the lobe that is just in front of the nose, and inion means we are going to have in the back. So over that distance, actually, what is the location of the electrode means that is obtained by observing the distance of either 10% distance or 20% distance. So that this is a that is an international the system that is used for the electrode placement, 10-20 electrode system. So this is the top view. This is the top view of the same. Okay, in this way, we are going to connect the various electrodes on the scalp. Okay, and based on that, we are going to measure the potentials. Based on that, we are going to measure the potentials. So here, these are the various EEG signals. So actually, based on the condition, based on the person condition, we are going to have various waves, various waveforms of various frequencies. If you observe, the person is in deep sleep. The person is in deep sleep. The frequency varies from 0.3 hertz to 4 hertz. The waveform will be like this, and that wave is called delta wave. Okay, if the person is in deep meditation, then the frequency range will be 4 to 8. That is said to be theta wave. And if the eyes are closed and the person is awake, then the frequency is 8 to 13, and that wave is said to be alpha wave. Similarly, if eyes are opened and thinking, the frequency range varies from 13 to 30. It is said to be beta wave. Similarly, for consciousness, it will be 30 and above. That is said to be gamma wave. So all these delta, theta, alpha, beta, gamma corresponding to the EEG signals. Okay, and actually here also by observing the abnormalities, by observing the changes in this EEG, we can find the various abnormalities in the patient. For example, this is an example how to identify, that is what is the EEG with the seizures. Epileptic seizures we are going to find in so many patients. To identify that epileptic seizures and all those things, we are going to have this type of analysis. So actually, the main thing is here we can analyze, we can perform the analysis of this either ECG signals or EG signals either in time domain or in frequency domain. If we consider the time domain signals, we are going to concentrate on what are the amplitudes of PQRS and what are the intervals of PQRS for healthy persons, and what are the amplitudes and this PQRS intervals for abnormal conditions. Similarly, we can consider this also. The other thing is we can consider in frequency domain. So by obtaining the frequency domain representation of these signals using any of the transformation technique, we can use fast Fourier transform or you can use wavelet transform. By using this type of transformations, we can convert these time domain signals to frequency domain signals, and so that we can have the parameters in frequency domain. And by actually in the nowadays, by comparing all these parameters either in frequency or time, and by applying the machine learning applications, there is a large scope for the research work in this domain, either for ECG signals or EEG signals. For biomedical signals, by obtaining the parameters either in time domain and frequency domain, we can apply various machine learning tools to analyze these signals, and so that to identify the abnormalities in ECG and EEG signals. Okay, this is a brief introduction for this, and how to proceed for what are the ideas for this research scope. Actually, what we can perform either in time or frequency domain. Frequency domain means we can apply wavelet transforms. There are number of journals or papers are there based on the applying the wavelet transforms for the analysis of this ECG or EEG signals. Thank you one and all for giving this opportunity, and CRM.
Yes. Thank you, madam. Thank you.
Whether you may satisfied with we are expecting more, or think and next time I am going to give another detailed analysis for the detection of these signals.
No problem.
Okay, thank you.
Yeah. So participants, if you have any doubt, please you can ask. Participants. Thank you, madam, for your effort bringing the biomedical signals to this platform, madam. So thank you, madam. So I thank you all the participants. So okay. With this, I close the session. Thank you.